All right, we are going to start. Pedigree analysis in human genetics. You guys went over pedigrees a little bit last week in chapter 3. Just kind of the general symbols, circles and squares, stuff like that. Here we're going to get a little more in-depth on pedigrees and what they look like if a certain disease or disorder or trait runs in a family. So this is chapter 4. Pedigree analysis can reveal the dominant or recessive inheritance pattern of a trait. It can also reveal the chromosomal locus of the gene in question. Remember, genes have certain locations on chromosomes, and those locations are called a locus or loci if you're just talking about one. Pedigrees are used to predict the risk of inheriting a specific trait, a risk to a fetus or future child of affected parents, or a risk of a family member developing an adult onset disorder. Some disorders are genetic, but they don't come over a person until they're actually adults. Huntington's disease is one of those, and we will talk about that. So we learned about dominant and recessive inheritance patterns of traits in the last chapter. Dominant is the stronger one. It will mask the recessive one if it's present. And this happens for a lot of different diseases too. Basic patterns of Mendelian inheritance. We went over Mendelian versus non-Mendelian in the last chapter. There's autosomal recessive, autosomal dominant, X-linked dominant, X-linked recessive, and also Y-linked. You guys did a lab on recessive, dominant, X-linked. You did X-linked recessive because it was about color blindness. And those are just some examples of pedigrees from those different types of patterns. All right, pedigree analysis used to be done by hand, but like everything else, it's become computerized. There's software that can automatically generate pedigrees. It will exclude patterns of inheritance that are inconsistent with pedigree data. So sometimes it needs to be looked at more closely, even when it's generated by a computer. And if only one pattern of inheritance is supported by the data, that is the accepted pattern of inheritance of that trait. Meaning if it looks like a duck and quacks like a duck, it's probably a duck. The process takes a lot of trial and error. If you're analyzing a pedigree, the first you try to rule out patterns of inheritance that are inconsistent with the pedigree. So for example, only males carry a Y chromosome. For traits controlled by a gene on the Y, only males will be affected. So if you're looking at a pedigree and there are circles filled in, the circles are females, it means it's not going to be a Y-linked disease because females don't have Ys. So you go through it and you look at the patterns and you rule stuff out. The Y-linked is easy to pick out because only males are affected. We will talk about that. We'll see some examples. Disorders with autosomal recessive patterns. Pedigrees of autosomal recessive disorders will have the following patterns. Affected individuals with rare traits have unaffected parents, can have unaffected parents. Each parent must transmit the mutant or recessive allele for the child to be affected. So this happens when the parents are heterozygous. And they have a 25% chance of passing both recessive alleles onto their child. So if you remember, the half and half are called carriers, and they are the heterozygotes for the disorder. And if you see a picture of a pedigree with circles and squares that are half filled in, an autosomal recessive is the inheritance pattern. This is what you guys are going to be doing on your quiz and on your test. You're going to be looking at these pedigrees and picking out the inheritance patterns. Pedigrees of autosomal recessive disorders will have the following patterns. Here's some more indications. The risk of having an affected child when both parents are unaffected heterozygotes is 25%. That you can see in the Punnett square. You can see parent 1 is heterozygous, parent 2 is heterozygous. There's the Punnett square between them. You have 25% chance that their child has the disease. All children of affected parents are affected. So if you only have recessive alleles to pass on, your child will end up with that recessive allele. And the trait is expressed equally in both males and females. So it's not X or Y related. It's in the autosomes somewhere. There's a whole table of different disorders and what happens when a person has these autosomal recessive disorders. Albinism, absence of pigment in hair, skin, and eyes. That's when we call them albinos. Cystic fibrosis is another common one. Sickle cell anemia is pretty common. So there's all kinds of them. And then the number on the side, we'll talk about that at the end. Disorders with autosomal dominant inheritance patterns. Remember, for regular traits, if a person is heterozygous, the dominant phenotype will be shown. And it's the same for dominant disorders. Heterozygotes will show the disorder. So if you have a normal parent and an affected parent, like in the Punnett square right there, that big A, that capital A, is what carries the disease. So if the children end up with the capital A from the affected parent, they will have the disease. They only need one because it's a dominant disease. The dominant will mask the recessive, and in this case, the dominant is the disease. The recessive is healthy. The healthy gets masked or hidden. Continuing on with the dominant disorders, a pedigree for a family affected with dominant disorders will have the following patterns. Every affected individual has at least one affected parent. Since most affected individuals are heterozygous, 2 affected parents could have an unaffected child. We'll see how that happens with dwarfism in a minute. Heterozygous individuals with a homozygous recessive spouse have a 50% chance of having an affected child. That was the Punnett square in the previous slide. There are no carriers in the pedigree. You either have the disease or you don't. And the trait is present equally in males and females. These diseases are not X or Y related. They can affect either gender. This is autosomal dominant pedigree. You note there are no carriers in the pedigrees of dominant disorders. They affect males and females equally, and they either have the disease or they do not. And then there's examples. Achondroplasia is the first one, and that's dwarfism. That's the one we're going to talk about. But there's quite a few others. But if you have the dominant allele, you have the disease. You only need one. There are questions in the quiz and in the test about the disorders, so make sure you take note of these charts. An example of an autosomal dominant trait, the heterozygous phenotype is typically less severe than the homozygous dominant when it's a dominant disease. In some cases, like achondroplasia or dwarfism, the homozygous dominant genotype is fatal. That baby does not survive if they get both dominant alleles. But two affected parents can have an unaffected child. So two dwarfs can get together and have a normal-sized child. And this is why. Because this disease is a disease of heterozygotes. If they're heterozygous, they end up with the disorder. If they're homozygous dominant, the disorder kills the fetus. And if they're homozygous recessive, then they have normal height. Sex-linked inheritance involves X and Y chromosomes. So genes on sex chromosomes have distinct patterns of inheritance. Males have an XY as their sex chromosomes. And they have a 50-50 chance of passing on either chromosome. If during fertilization they pass on their X, they will have a daughter. If during fertilization males pass on their Y, they will have a son. If you think of the word boy ends in Y. If you have a Y, you are a boy. Females have two X's. They only have X's to pass on. So they pass the X chromosome on to all of their children. So it's actually the father that determines the gender of a child, because the father has two different ones and they have a 50-50 chance of passing on either one. Most genes on the X chromosome are not found on the Y chromosome. So this is just a Punnett square of how gender comes about. Males have XY. They can pass on either one. Females have XX. They got a 50% chance of having a boy or a girl. All right, females can be heterozygous or homozygous for X-linked trait because they have two X's. So in a Punnett square, we draw both the X and the allele. We draw them as a superscript. That just means they're a little bit above the X. So the first one, she would be homozygous dominant for color blindness, which means she is normal. The second one, on the second square on the right, she is heterozygous for the colorblind allele, which means she's a carrier. She carries that colorblind allele. She's not colorblind herself because the dominant B masks the diseased one, but she is a carrier and she can pass it on to a child. We went over this a little bit last chapter when you did the lab. Or you should have gone over the presentation while you were doing the lab, and this was in there. Males only have one X chromosome, so they are called hemizygous for X-linked traits. And hemizygous is a condition which only one copy of a gene or DNA sequence is present in diploid cells. Males are hemizygous for most genes on sex chromosomes because they have one X and one Y. So if it's an X-linked trait, they only have one allele that goes with it, and they will exhibit whatever that allele is. So males carrying one X-linked recessive allele express the recessive phenotype because they only have one X. You don't link the allele to the Y because it's not linked to the Y. That's why it's called an X-linked trait. So in his Punnett square on the left, he'll be normal. That boy on the left will be normal because he has the dominant allele. On the right, that boy will be colorblind because he only has one allele for the colorblind gene, and it is recessive. So that recessive allele will show. Only a few X-linked traits show dominant inheritance patterns. Most of them show recessive, but there are a few dominants. Affected males will pass the trait on to all of their daughters, who will be similarly affected, but none of their sons. Heterozygous affected females will pass the trait on to half of their children, both males and females, who will be similarly affected. And there are, on average, twice as many affected females than males because females have two X's, and if either one of those X's is dominant, they will show the disorder. So if you look at a pedigree and you see that twice as many females are affected as males, then it's most likely a dominant inheritance pattern, an X-linked dominant inheritance pattern. So you can see the affected males produce all affected daughters, but no affected sons, because they only have the one X to pass on, and if the one X that they have is linked to a diseased allele, that's what they pass on to their daughters. And affected females transmit the trait to about half their children because they have the two alleles. They can pass on either the X with the good allele or the X with the bad allele. So twice as many females as males are affected with the trait. Disorders with X-linked recessive inheritance patterns are more common. There's over 850 identified X-linked recessive traits. Hemizygous males and homozygous recessive females are both affected. And it's much more common in males, like I said, because they don't have a second X to mask the disease. And in some rare traits, they're observed exclusively in males because they don't, I guess I already said that. Affected males pass the trait on to all of their daughters, but none of their sons. Daughters of affected males are heterozygous carriers and unaffected themselves. And then sons of heterozygous females have a 50% chance of inheriting the trait and being affected. And there's an example. This one should be recognizable, this type, because the carriers are marked a certain way. Homozygous males are affected and transmit the trait to all of their daughters. Their daughters become carriers. And the phenotypic expression, meaning what is seen on the outside, is much more common in males than in females. So only females can be carriers because they have the two X's. But males will actually have the disorder more often. And you can see that in the actual pedigree. So this pedigree should be easy to recognize in your quizzes. Color blindness is an example of an X-linked recessive trait. Defective color vision caused by reduction or absence of visual pigments. There's three forms. You could be red-green colorblind or blue colorblind. And about 8% of the male population in the United States is affected. So where most people can see the oranges and greens and everything in that bottom picture, that top picture just kind of looks gray. And then here's a test for you. Let's people with normal color vision see the number. You should see the 15 in the chart. Those who are colorblind cannot see the number. So if you can't see the number, there is a chance you are colorblind. And there's actually a company, I should have linked the video, I can link the video later. Valspar is a paint company, but they're also made glasses for colorblind people so they can actually see colors. And it's actually a really sweet video. So there's examples of X-linked recessive traits. Color blindness, the red-green, hemophilia, muscular dystrophy, those are X-linked traits. And then genes on the Y chromosome, also pedigrees that should be really easy to pick out. So only males have Y chromosomes, boys have Ys. Genes are passed directly from father to son. All Y-linked genes are expressed because there is only one. They're also homozygous for genes on the Y chromosome. And to date, there's only 60 Y-linked traits that have been identified, many of which are involved in male sexual development. And then there's your Y-linked inheritance pattern. Like I said, it should be easy to pick out because only boys are affected. All the squares are filled in, none of the circles are. Males only have that one Y to pass on, so all of their sons are going to be affected. Non-Mendelian maternal inheritance. Like I said, there's not all traits follow Mendelian inheritance patterns. So we call them non-Mendelian. Mitochondria contain DNA that encodes proteins involved in the conversion of food energy into ATP. It's what powers all of our cells to be able to do work. Genetic disorders in mitochondrial DNA are associated with defects in energy conversion. So somebody who is chronically tired might have something wrong with the mitochondria in their cells. Their energy is just not being converted properly. They might also just be old because old people are tired. Mitochondrial inheritance. Females pass mitochondria onto all of their children through the cytoplasm of the egg. Sperm do not contribute cytoplasm or mitochondria to offspring. So all children of females with mitochondrial mutations will be affected. But only the daughters will continue to pass this trait on to their offspring. So men can be affected, but they don't pass it on to their offspring. And that's what it looks like in the pedigree. Pedigree showing the pattern of inheritance associated with mitochondrial genes. Both males and females can be affected, but only the females transmit it to their children. So you can see in generation 2, number 1 is affected, but he doesn't pass it on to his children. Same with child number 5. Doesn't pass it on to his children. But if the mother has it, then all the kids have it. And there's some examples of mitochondrial disorders. They're not very common, but they do happen. And there is an online catalog of human genetic traits. It is free. I linked it there if you want to look at it. Online Mendelian Inheritance in Man is what that stands for. It's an online catalog of all known human genetic traits from the Human Genome Project when we map them all. It's part of a larger series of integrated databases. The genetic traits are described, cataloged, and assigned a MIM number. And that's in the previous, the MIM number is on the right. That's where that comes from, is this database. And it's freely available to anybody who wants to look up this information. It's not just for scientists. Many factors can affect the outcome of pedigree analysis. Phenotypes are often age-related. For example, Huntington's disease is a dominant disease, but symptoms don't show up till between the ages of 30 and 50. So it's well past the childbearing age, so a lot of people that end up with Huntington's disease have already had children and passed it on to them. Penetrance is a fun vocabulary word. Describes the probability that his disease phenotype will be present when the genotype is present. And if the probability is less than 100%, the trait shows incomplete penetrance. Like I said, for the mitochondrial diseases, it is 100% that the mother will pass it on to all of her children. That's the probability that she will pass it on. It's called penetrance. But if it's only a 50% probability, like just a regular recessive disorder, or a 25% probability, we call that incomplete penetrance. And expressivity, the degree of phenotypic expression. Traits that do not show uniform expression have variable expressivity. Common recessive alleles can enter a family pedigree and produce patterns that resemble dominant inheritance. So dominant alleles aren't always common. Sometimes recessive alleles are common. And because it's more common, people think, oh, it's very common, it should be dominant. That's not always the case. Sometimes it's just common because it's common. Figure 422, I'll show you in a minute, is about penetrance and expressivity. It's about blood types. Well, this one's about camptodactyly in a family with both variable penetrance and expressivity. Camptodactyly is, you can see the picture, with immobile bent little fingers. So this pedigree is always fun because fully shaded symbols indicate members with 2 affected hands. Sometimes the left hand is affected, and then you just shade the left side. Sometimes the right hand is affected, then you just shade the right side. And then you'll see there is no penetrance in individual 3-4, even though he passed the gene to all of his children. Variable expressivity includes several phenotypes, including no phenotypic expression, meaning you can't see it at all on the outside. Expression in one hand or both hands. That's why it's called variable. It's not the same every single time. It's different. So that's the pedigree for incomplete penetrance and variable expression. Sometimes it's shaded in on the right if the right hand is affected. Sometimes it's shaded in on the left if the left hand is affected. It's fully shaded in if both hands are affected. And if you can't see it at all, the gene might still be there, but it's just not shaded in. And then common recessive alleles can enter a pedigree from outside the family and thus appear dominant. Blood type is one of those. O positive blood is very common. It is also recessive. Many people have type O blood. As a result, the inheritance of this allele produces a pseudo-dominant pedigree, with members of each generation showing the trait and about half of the offspring in the next generation. I said, just because it's common doesn't mean it's dominant. And blood typing is one of those where the recessive is more common. In nature, it's called the wild type phenotype. Whatever is more common. And yep, that is it. That is it for chapter 4. It's a short week, so I only have one assignment this week. Putting together pedigrees, simulating a pedigree. Any questions? Yes, no? Tell me yes or no in the chat box.
No.
No? Okay. That's it for today. Like I said, the work from last week is still open. Today's the last day to do it. You will get points off for it being late, but you can still do it up to today. So finish what you need to finish and work on this week's work. Bye. I will see you next week. I won't see you till next week. So Monday.
Bye. Have a good day, Mrs. Ruiz.
Have a good day. I'll see you Monday.
See you Monday.
Have a good one. Bye, Julian. Bye, Jasmine.
Have a good day, Miss Ruiz.
Have a good day, Adrian. Take care, Maria.